The core facilities offers slide scanning as a service. Our Zeiss AxioScan Z1 is capable of both bright field and fluorescent whole slide scanning. It is ideally suited to image thin tissue sections, but can also be used to image cell monolayers. The AxioScan is equipped with 5 objective lenses, from 2.5x to 40x. The bright field camera is a Hitachi 3-chip color camera, which produces 8-bit RGB images. The fluorescence camera is a Hamamatsu Orca Flash, which produces 16-bit monochromatic images. The fluorescent light path uses a Colibri 7 LED light source capable of excitation from 385 nanometers to 630 nanometers. And there are 5 sets of emission filters. To view the complete specifications of our slide scanner, please check the RCF website. For external users, please refer to our fee schedule located on the website. Both of these will be linked in the description below. Preparation of slides for scanning. There are 4 steps that you need to follow to ensure that your slide scanning goes smoothly. Following these instructions will ensure that your images will be clear and in focus. Number 1. Make sure your slides are clearly labeled. Here is an example of a good label. The words are written on a white background in black font. Here is an example of a poorly labeled slide. The background is clear, and there's not enough contrast in the writing to be legible. Number 2. Your mounting media must be fully dry. You don't want to inadvertently damage or destroy your sample, so make sure they can't move around, as this is not their dancing competition. Number 3. Ensure all surfaces are clean and don't have any residue. This could impact the visibility of your scan, and we don't need to see what you were snacking on earlier. Number 4. Do not have large amounts of mounting media at the edges of the cover slip. Excess dried up media on the surface can interfere with results and damage the machine. If you follow these 4 steps, this will ensure that your images will be clear and in focus. Scan settings. For fluorescent samples, the optimal scan settings for each of the fluorophores must be determined. In order to do this accurately, the positive and negative control slides must be indicated. Be sure to indicate which slide and tissue section contains your positive control for all fluorophores. This can be done by drawing a plus and an arrow on the slide. You may also indicate your negative control slide and a section in a similar fashion, using a minus sign and an arrow. Note that fluorescent scan settings are kept constant for your dataset. You may find the scan settings on the requisition form or in the software on the info tab. The scan settings contain all the hardware settings for the focus strategy, as well as the data acquisition, including light path, LED, exposure time, etc. Dropping off samples. Use a slide box and not a paper folder. This will ensure that your slides are kept safe and won't jump ship. You can keep your samples in room temperature or 4 degrees. Typically, fluorescent slides are stored in 4 degrees. Thankfully, we have a mini fridge for that. Tape your requisition form to the box, or put it inside the box. If you have a hard drive, make sure it's not just a USB stick. Our file sizes are typically several hundred megabytes to a few gigabytes per slide. Finally, when you drop off your samples, you may send an email. This is optional, as we tend to check the area daily. So, your slides are done. Now what? The file format is CZI. Keep this file format for your raw data. You can view your data with Zen Lite. It's free to download for a PC. The link to this is included below. As the data is quite large, you may need to book one of the available workstations if you require more computing or graphics power. For analysis workstations and software, we have the Halo PC in room 778, QPath in room 552, and Zen Lite, which is available on all workstations, in room 552. For the QPath, since this is an open source software, you may also use your own PC, although you may need the computing power of one of our workstations. You can book these workstations through the RFBMS. Please be sure to fill out the slide scanning requisition form as follows. Please fill out all the areas. Note that the specialists will fill out all the gray areas. Use one form per experimental set. This is important for fluorescence datasets. If you have different stainings, you will need a new form for each different staining. Keep this form for your records after scanning, as there will be important information on it about how your data acquisition was completed. Dirty slides will not be scanned, and fluorescent samples should be carefully outlined in black marker and should not include areas without tissue, as described earlier. Please write your name, lab, email, and the date submitted. You'll also include your number of slides, whether it's a cell or tissue section, tissue thickness, whether it's FFPE or cryo section, and whether you want to transfer your data to the analysis PC. Note that if you do not indicate yes here, we will no longer automatically transfer your data to the analysis PC. If using bright field scanning, check bright field and skip the fluorescence area. Then, indicate what magnification you'd like to scan. 20x is going to be ideal for about 95% of situations. Typically, 20x is sufficient for scanning if you want to look at cells or nuclear translocation and even some subcellular compartments. 40x 0.95 NA scanning is typically for in situ hybridization. Don't choose 40x unless you really need to, or else your data sets will be really large and hard to manage unnecessarily. If doing fluorescence, check fluorescence. Then, indicate which fluorophores your sample is stained with. For example, if you're using DAPI and it's excited by UV, check UV for excitation of DAPI. For fluorescence, it's very important to specify which slide has your control. If your slide has more than one section, please indicate which section. If you have special instructions, you may include them here. For example, if you need a Z-stack or any processing, or for example, your positive signal is located in a certain area, the specialist will know where to look for your signal. Once your scan is complete, the specialist will fill out the date that it was scanned, and they will also indicate the scanning profile. The scanning profile will contain all the settings the specialist set up for determining the focus map and acquiring your data. Be mindful that the focus map settings do not influence your data acquisition. The exposure time for each of your fluorophores will be indicated here in milliseconds. If you would like to use the same scanning profile for another set of slides at a later date, please indicate in the special instructions which scanning profiles you'd like to use. This means that we'll set up the exact same exposure times. So, do not indicate that you want to use the same scanning profile if the exposure times may not be optimal. It's easier for us to set up a new scanning profile in that case. That concludes our slide scanning instructional video. If you follow all of the steps, your slides should be scan-tastic.